Nucleotides are the monomers that make up nucleic acids, including DNA and RNA. A nucleotide consists of a sugar molecule attached to a phosphate group and a nitrogenous base. As I mentioned in my previous video titled Nucleic Acids, the nitrogenous bases are further classified by the number of rings they contain. Purines such as hypoxanthine, guanine, and adenine contains 2 fused rings. Pyrimidine such as cytosine, uracil, and thymine only contain a single ring. Nucleotide biosynthesis involves 2 pathways: the de novo pathway that synthesizes nucleotides from their metabolic precursors, and the salvage pathway that recycles the free bases and nucleosides released from nucleic acid breakdown. I'll first cover the de novo pathway of nucleotide biosynthesis. Note that nitrogenous bases are not synthesized separately and then attached to the sugar ribose, but instead build up one or few atoms at a time. The sugar phosphate backbone is derived from glucose 6-phosphate, which is an intermediate of glycolysis. First, glucose 6-phosphate enters the pentose phosphate pathway and undergoes a series of oxidative steps to produce ribose 5-phosphate, which is subsequently converted to 5'-phosphoribosyl 1-pyrophosphate, abbreviated as PRPP, by the enzyme PRPP synthetase, with ATP as a phosphate group donor. PRPP serves as a precursor to the de novo biosynthesis of both purine and pyrimidine nucleotides. PRPP also serves as a precursor to histidine biosynthesis in bacteria, which I've covered in my video on amino acid biosynthesis. In the first committed step of de novo purine nucleotide biosynthesis, an amino group donated by glutamine is attached to carbon 1 of PRPP by the enzyme glutamine PRPP aminotransferase to produce 5-phosphoribosylamine, and the purine ring is subsequently built up on this structure through a series of 10 steps. The first intermediate with a complete purine ring is inosinate or IMP. Nitrogen 1 is derived from aspartate, carbons 2 and 8 are derived from N10-formyl tetrahydrofolate, abbreviated as FH4. Nitrogens 3 and 9 are derived from glutamine, carbons 4, 5, and nitrogen 7 are derived from glycine, and carbon 6 is derived from carbon dioxide. IMP serves as a precursor to other purine nucleotides. Carbon 2 of IMP can undergo oxidation to produce xanthylate or XMP, which is coupled to the reduction of NAD+ to NADH catalyzed by IMP dehydrogenase. XMP can then be converted to guanylate or GMP by XMP glutamine aminotransferase, which replaces the ketone group at the second carbon with an amino group donated by glutamine. This reaction is also coupled to the hydrolysis of ATP to AMP and inorganic pyrophosphate. IMP can also undergo 2 reactions to produce adenylate or AMP. First, adenylyl succinate synthetase condenses IMP with aspartate to produce adenylyl succinate, which is coupled to the hydrolysis of 1-GTP to GDP and inorganic phosphate. Next, adenylyl succinate is cleaved by adenylyl succinate lyase to form fumarate and AMP. From these reactions, we can see that the extra amino group on carbon 2 of GMP is derived from glutamine, while the extra amino group on carbon 6 of AMP is derived from aspartate. On the other hand, de novo pyrimidine nucleotide biosynthesis starts with the condensation of glutamine and bicarbonate to produce carbamyl phosphate, which is catalyzed by carbamoyl phosphate synthetase 2, abbreviated as CPS2. Note that CPS2 is an isocyme of CPS1 of the urea cycle, which means that they catalyze the same reaction but are regulated differently. Carbamyl phosphate then condenses with aspartate by the enzyme aspartate transcarbamylase to produce N-carbamoyl aspartate, which undergoes dehydration reaction to form a closed ring known as L-dihydroorotate. In eukaryotes, the first 3 enzymes CPS2, aspartate transcarbamylase, and dihydroorotate are part of a single trifunctional protein known as CAD. Next, dihydroorotate is oxidized to the pyrimidine derivative orotate, which is coupled to the reduction of NAD+ to NADH. Then the ribose 5-phosphate side chain provided by PRPP is attached to orotate to form orotidylate, also known as OMP, which is subsequently decarboxylated to uridylate or UMP. From these reactions, we see that nitrogen 1 and carbons 2 through 4 are derived from aspartate, nitrogen 5 is derived from glutamine, and carbon 6 is derived from carbon dioxide or bicarbonate. UMP is phosphorylated by kinases to form UTP, which can be converted to CTP by the enzyme cytidine synthetase, which is coupled to the hydrolysis of 1 ATP molecule and involves glutamine as the donor of amino group attached to the 4th carbon. Deoxyribonucleotides, the building blocks of DNA, are derived from corresponding ribonucleotides by direct reduction at the 2' carbon atom of ribose to form 2' deoxy derivative. However, the substrate of this reaction involves ribonucleotide diphosphate. UMP can be phosphorylated to UDP by UDP kinase, with ATP as a phosphate donor. CTP can be hydrolyzed to CTP by CTPase. Ribonucleotides UDP and CTP can then be converted to deoxyribonucleotides, DUDP and DCDP by the key enzyme ribonucleotide reductase. The reduction of the ribose portion of a ribonucleotide diphosphate to 2' deoxyribose requires a pair of hydrogen atoms, which are ultimately donated by NADPH through either glutaredoxin or thioredoxin. DUDP and DCDP can then be phosphorylated by diphosphate kinase to form DUTP and DCTP, with ATP as a phosphate donor. DUTP serves as a precursor to thymidylate or DTMP. DCTP can be converted to DUTP by deaminase. DUTP can undergo hydrolysis to produce DUMP and inorganic pyrophosphate. Thymidylate synthase catalyzes the transfer of 1 carbon unit from N5N10 methylene tetrahydrofolate to DUMP, producing DTMP and 7,8 dihydrofolate, which can be reduced to tetrahydrofolate by dihydrofolate reductase, coupling to the oxidation of NADPH to NADP+. Tetrahydrofolate can then be reconverted back to N5N10 methylene tetrahydrofolate by serine hydroxymethyltransferase, abbreviated as SHMT, which is part of serine and glycine catabolism and anabolism and requires the cofactor pyridoxal phosphate or PLP, as mentioned in my previous video about amino acid metabolism. De novo nucleotide biosynthesis is regulated by feedback inhibition. PRPP synthetase is inhibited by ADP. Glutamine PRPP aminotransferase is inhibited by all 3 purine nucleotides, including IMP, GMP, and AMP. And adenylosuccinase synthetase is inhibited by AMP. For pyrimidine biosynthesis, CPS2 is inhibited by UTP and activated by PRPP, while aspartate transcarbamylase is inhibited by CTP. Many chemotherapeutic agents target enzymes in the nucleotide biosynthetic pathways, as cancer cells have greater requirements for nucleotides as precursors of DNA and RNA, and are more sensitive to inhibitors of nucleotide biosynthesis. Glutamine aminotransferases are inhibited by glutamine analogs such as azathioprine and acifezin. Thymidylate synthase can be inhibited by fluorouracil, abbreviated as 5FU, through mechanism-based inhibition. Finally, dihydrofolate reductase can be inhibited by methotrexate and aminopterin, which are folate analogs. On the other hand, the antibiotic trimethylprim inhibits bacterial dihydrofolate reductase nearly 100,000 times better than mammalian enzyme. Therefore, it is used to treat certain urinary and middle ear bacterial infections. In the last part of the video, I'm going to cover the salvage pathway in which free nucleobases are recycled to build nucleotides. Hypoxanthine guanine phosphoribosyl transferase, abbreviated as HGPRT, catalyzes the condensation of free hypoxanthine, xanthine, and guanine with PRPP to produce inosinate, xanthylate, and guanylate. Genetic defect in HGPRT will result in Lesch-Nyhan syndrome, which causes coordination problems and intellectual deficits. Free adenine is salvaged in a similar fashion by the enzyme adenosine phosphoribosyl transferase, abbreviated as APRT. Nucleotide biosynthesis will be covered in a future video.